Good morning, students. So we'll begin up with today's class. That's your respiration in your lungs. Okay. Now, when we use a term respiration, okay, the respiration can be defined as your oxidative process. That means when I use a word called as oxidative, always there is a particular oxygens. Am I clear to you? So during which food materials undergo oxidation, that's nothing but your oxygen is added to the food and gets converted into carbon dioxide, water and your energy. Okay. So whenever during the respiration, whatever the food you intake, or whatever is the particular organic compound you intake, okay, always to that particular food or anything, the oxygen is definitely required, okay, and then it will get converted into what and what makla, it will get converted into carbon dioxide, water and your energy, okay. Now in the respiration you have two types of respiration: external respiration and internal respiration. What are all makla? External respiration and internal respiration. Now, whenever I use a term called as external respiration, it is an exchange of gases, oxygen and carbon dioxide, between organism and its surroundings. Okay, I repeat again. ಏನಂತ ಹೇಳ್ತಾ ಇದ್ದಾರೆ ಅಲ್ಲಿ? It is an exchange of gases. ಯಾವ್ಯಾವ್ದು? Oxygen and carbon dioxide. Between what and what? Between the organism and surrounding. Now you are there and the surrounding. Yes or no? So between you and the surrounding, there is an exchange of oxygen as well as your carbon dioxide. Okay. Now moving ahead, internal respiration is an exchange of a respiratory gases between tissues, cells and surrounding environment. Andre idu nimma ಒಳಗಡೆ, within inside you. Okay. So here what is happening? There is an exchange of a respiratory gases between what and what makla? Between the tissues. Okay. So inside the tissues of you and the surrounding environment. So within your body. Am I clear to you? So the cells within you and the environments in your body. Okay. And plus outer environment also. Okay, children. So this is how, what do you mean by internal respiration? External and the organism and surrounding, whereas the respiratory gases between the tissues and the surrounding environment and then ನಿಮ್ಮ body ಒಳಗಡೆ. Am I clear to you? So that's called as your internal respiration. So these are the two types of your respiration. ಯಾವ್ಯಾವ್ದು ಮಕ್ಕಳೇ? The external respiration and your internal respiration. Okay. Moving ahead, why is the respiration important? What is the significance of the respiration? Okay, so respiration plays an important role in what? In the particular plants, also in the particular animals, but now we are speaking a significant role in the plants. Okay, so in the particular, during the particular process of the respiration, it releases energy, okay, which is consumed in various metabolic particular processes necessary for a life of a plant. Okay, so what is it, children? In this particular thing, usually the respiration releases the energy. Okay, so during the respiration, whenever there is an oxidation, there is an energy release. Am I clear to you? So that is a very important point. Am I clear to you? Now why is this energy necessary? This energy is used in various metabolic processes. It might be your glycolysis, Krebs cycle, your etc, okay, so on and so forth. So for most of the things you do require energy. So in all these things, what is it, children? The particular respiration plays an important role. Moving ahead, second one, energy produced can be regulated according to the environment of all activities. So whatever the energy is released, the same point, even it is telling you, what is it telling you? It is telling you like it can be used in a various metabolic processes. Am I clear to you? So energy produced can be regulated according to the requirement of all activities. ಏನೇ ಎನರ್ಜಿ ಪ್ರೊಡ್ಯೂಸ್ ಆದ್ರೂ ಮಾತ್ರ, ಸೊ ನಮ್ಗೆ ಏನಕ್ಕಾದ್ರೂ ಅಂದ್ರೆ ಪ್ಲಾಂಟ್ ಏನ್ ಬೇಕಾದ್ರೂ ಯೂಸ್ ಮಾಡೋದಕ್ಕೂ ಯು ರಿಕ್ವೈರ್ ದಿಸ್ ಎನರ್ಜೀಸ್. ಓಕೆ, ನಾ ದ ನೆಕ್ಸ್ಟ್ ಒನ್, ವಾಟ್ ಇಸ್ ಇಟ್? It converts insoluble foods into soluble form. Okay, so insoluble food na ಏನ್ ಮಾಡ್ತಾ ಇದೆ ಮಕ್ಕಳೇ? It is converting into the soluble form. ಯಾವುದು? Respiration. Okay na? So during the respiration, what is happening? It is converting your insoluble foods into soluble foods. Okay. Now, intermediate products of cell respiration can be used in a different metabolic pathway. Okay. So intermediate ಅಂದ್ರೆ ನಿಮಗೆ ಇವಾಗ ಎಕ್ಸಾಂಪಲ್ ಪೈರುವೇಟ್ ಆಗಿರಬಹುದು. Okay, so if at all the particular pyruvate which is formed, it is also used in Krebs cycle. So like that you have PGA also. Yes or no? So even in the PGA, it is used in your Calvin cycle if at all you are seen about it. Am I clear? So any intermediate product whichever is formed, okay. So all those things are used in another metabolic processes, okay. So once again, I'm repeating what is it? The significance of respiration. First one, it releases energy which is consumed in various metabolic processes necessary for life of a plant, okay. Second one, energy produced can be regulated according to the requirement of all activities. Now, the next one is insoluble food is usually converted into the soluble form. Intermediate products, whatever is formed during the respiration, can be used in another metabolic pathways. Okay. Now, moving ahead. What's the difference between photosynthesis and respiration? Now, why did I put it up so that last class I had dealt with last week, if I'm not wrong. We had dealt with photosynthesis, so I thought it would be better for you to know about it. Okay, so photosynthesis occurs only in the chlorophyll containing cells of plants. Okay, whereas the respiration occurs in all plants and animals. Okay, so this takes place only in the light, whereas respiration, unlike photosynthesis, takes place both light as well as in the dark. Okay, so respiration, during respiration potential energy is converted into kinetic energy, but in photosynthesis radiant energy is converted into potential energy. Next one, sugar, water, oxygen are the products of photosynthesis, whereas for the respiration, carbon dioxide and water are the products. Okay. Next one, photosynthesis synthesizes the foods, whereas the respiration oxidizes the foods. Photosynthesis, carbon dioxide and water are the raw material. So initially you give water and carbon dioxide, where your carbon dioxide getting reduced to your carbohydrate plus your water is released, oxygen is released along with the energy. Whereas here, oxygen and the food molecules are the raw material where it will get converted into carbon dioxide and water. Okay. Next up, photosynthesis is endothermal process. Idu nimge exothermal process. Okay. So photosynthesis stores energy. Okay. This releases the energy. Next one, it includes the process of hydrolysis and carboxylation. Hydrolysis and the splitting, carboxylation and the fixing of carbon dioxide. Whereas here, it involves your dehydrolysis, removal of the water, and decarboxylation, removal of the carbon dioxide. Results in increase in the weight, results in the decrease in the weight. Okay, it is your anabolic process. This is your catabolic process. Requires cytochrome. It also requires the cytochrome. Okay, so usually these are the particular differences between your photosynthesis as well as your particular respiration. Now, after this, characters and cell respiration and the combustion. Okay, so what are the particular characteristic of a cell respiration is the next point. Children, when it comes to the characteristics, okay, so first one is your nature of process. What is it, Makla? Nature of process. Now in this cell respiration, the biochemical and the stepped process it is. Okay, so combustion, it is a physiochemical and your spontaneous process. Usually, Makla, your respiration, of course, it's a chemical process and it is a spontaneous. Am I clear? Next up, what is the occurrence of the cell respiration? It always occurs inside the cell. Okay, so combustion, it is a non-cellular. Control, cell respiration is always a biological control, and combustion you cannot, it is usually an uncontrollable. Am I clear? So, ಇದರಲ್ಲಿ difference ಕೊಡ್ತಾ ಇದ್ದಾರೆ. Hope you are understanding. One is cell respiration, another one is your combustion. Okay. So, energy release is in steps, whereas large amount of energy is released at a time. So, combustion gu respiration gu nimge differences ಕೊಡ್ತಿರೋದು ಮಕ್ಕಳೇ. Respiration is one what we go through. Am I clear? To combustion, it can be a particular, what to say, a spontaneous point. Am I clear to you? Andre sudden ಆಗಿ ನಿಮಗೆ ಒಂದು combust ಆಗಬಹುದು ಅಥವಾ the energy which is combust. So that is the difference between these two. Okay, ಮತ್ತೆ ಒಂದ್ಸಲಿ ಹೇಳ್ತಾ ಇದೀನಿ, the nature of process. So cell respiration usually is a biochemical and it is a stepped process. Usually takes place inside the cell. It is biological control, means we control it, the system controls it. Energy release is usually released in a stepwise. Temperature, it is always within the limits. Am I clear to you? So light, no light is produced over here. Okay, because during respiration there is no light. Enzymes are controlled by respiration. So enzyme is usually controlling the process of respiration. Intermediates, a number of intermediates are produced. So ಮಧ್ಯದಲ್ಲಿ ನಿಮಗೆ ಎಷ್ಟೊಂದು ಪ್ರಾಡಕ್ಟ್ಸ್ ಬರ್ತಾನೆ ಇರುತ್ತೆ. Am I clear to? So that's about your cell respiration. So when it speaks about the combustion, it is opposite to your cell respiration. What? It is usually a physico-chemical spontaneous process. Site of occurrence is non-cellular, so it can happen outside the body also. So control is usually uncontrollable. Large amount of energy is usually released. Am I clear to you? So the temperature is very, very high. So whenever there is a combustion, in any combust madi, usually there will be a light produced, and it is definitely not controlled by your enzymes. Am I clear to you? And no intermediates are usually produced. Am I clear to you, Maka? So no intermediates are usually produced. Am I clear to you? So these are the few of the things about your respiration and combustion. So now let's speak only about the respiration. So it's a biochemical, as I told you. It is taking inside the cell, okay, where process is happening inside the cell, okay, and it's a biological control. Energy release usually is in a particular stepwise, okay. So always there is a limits. Am I clear to you? So limits illana process agala. Am I clear to you, okay? And no light is produced, definitely. Or else imagine children, whenever you're inhaling, so there should be always a light coming out. Am I clear to you? So there is no light coming out. And definitely your enzymes are always controlled by your enzymes. And of course there is a number of intermediates. You know about glycolysis and Krebs cycle. You have lots of intermediates produced in between. Now. Hello. Moving ahead, children, the next particular point, the glucose. Okay, so the particular process is happening in the two particular process, the fermentation. Next one is your particular fermentation and oxidative decarboxylation. Okay, so particularly children, your particular respiration happens in the two particular process, aerobic and your anaerobic. Am I clear to you? So under your particular anaerobic, am I clear to you? Usually in your anaerobic children, usually there will be a fermentation happening. Yen ಆಗ್ತಿರುತ್ತೆ ಮಕ್ಕಳಲ್ಲಿ fermentation ಆಗ್ತಾ ಇರುತ್ತೆ. Am I clear to you? So whenever there is a fermentation happening, there are two particular products produced. What are those? One is your ethanol, other one is your lactic acid. Am I clear to you, children? And of course, during your particular ethanol production, what will happen, children? Always you will have your carbon dioxide produced. What will happen? Usually you will have your carbon dioxide produced. Am I clear to you? So whenever there is an anaerobic respiration, children, so basically your glucose so taken inside. Am I clear to you? Whenever you take inside the glucose, so your glucose will undergo two particular process. Aerobic process and anaerobic process. So during your anaerobic, usually it will undergo a fermentation process. Am I clear to you? Where you will have your pyruvate. Alvendra. So pyruvate form ಆದ್ಮೇಲೆ glycolysis ಇಂದ ನಿಮಗೆ pyruvate ಬರುತ್ತೆ. Okay. So pyruvate in the absence of oxygen and in the presence of oxygen. Am I clear to you? So in the absence of oxygen, it is usually your anaerobic. In the presence of oxygen, usually it is aerobic. Okay. So during your anaerobic process, it will undergo your fermentation process. So in the fermentation, ಏನಾಗುತ್ತೆ ಮಕ್ಕಳೇ? Either you will have your ethanol produced, either you will have your lactic acid produced. Am I clear to you? So this is the thing. Once again, I repeat, you have your glucose. So the glucose so produced will undergo a process called as glycolysis. Okay, so glycolysis, which will result in what? It will result in your pyruvate. Ena result agutte makle idu pyruvate result agutte. Now what is the fate of a pyruvate? Now it is in the absence of oxygen and it is in the presence of oxygen. Am I clear to you? So usually in the absence of oxygen, what is happening? It is undergoing a process called as fermentation. What is it undergoing, makle? It is undergoing a process called as fermentation. Am I clear to you? So in fermentation, what happens? There are two things which are usually produced. One is your particular lactic acid, another one is your particular ethanol. Am I clear to you? So two things which are usually produced, lactic acid and ethanol, and of course you will have your carbon dioxide also produced. Whereas in the presence of oxygen, don't worry, you will be studying everything in detail, where it will undergo oxidative decarboxylation, Krebs cycle, electron transport chain. So when it goes all the three processes, ಏನಾಗುತ್ತೆ ಮಕ್ಕಳೇ, there will be 36 to 38 ATP. Okay, so 36 to 38. I will tell you about this little later. Okay, so usually it is 36, but sometimes it will also undergo 38. Am I clear to you? So usually it is 36 to 38 of ATPs which are usually produced. So glucose in the, so glucose will undergo glycolysis which will give rise to pyruvate. So pyruvate, in the absence of the oxygen, that will undergo your anaerobic process where it will result in a fermentation. So fermentation will result in your lactic acid formation and your ethanol formation. Am I clear to you? Whereas if it is undergoing your particular oxygen, that is your aerobic respiration, it will undergo your oxidative decarboxylation, Krebs cycle, electron transport chain, and your particular how many ATPs? 36 to 38 ATPs form ಆಗುತ್ತೆ. So this is the fate of your pyruvate. Either it will undergo your particular aerobic respiration or your anaerobic respiration. Okay, so let's start up the particular first cycle is due to the glycolysis. ಏನು ಮಕ್ಕಳೇ? Glycolysis. Okay, now in glycolysis what happens? You see children, the first one is nothing but your glucose. Okay, so glucose, okay children, will undergo the particular addition of the phosphate. How addition of the phosphate? ATP whichever is there, okay, will get converted into ATP. So ಏನಾಯ್ತು? Adenosine triphosphate got converted into adenosine diphosphate. So one phosphate was liberated. Yes or no? So now this phosphate will react with glucose to form glucose 6-phosphate. Okay, in the presence of an enzyme called as your hexokinase, which is the enzyme hexokinase. Am I clear to you? Next, your glucose 6-phosphate will undergo isomerization. What it will undergo? Isomerization to form what? Glucose 6-phosphate will undergo your isomerization to form what? Fructose 6-phosphate in the presence of an enzyme called as phosphoglucoisomerase. ಅರ್ಥ ಆಯ್ತು? ಅರ್ಥ ಆಗ್ಲಿಲ್ಲ ಮಕ್ಕಳೇ? Okay now, so the next particular phase, okay, what will happen? Your fructose 6-phosphate will again, okay, where there will be a particular, ATP will get converted into ATP, where again your one phosphate will be liberated. So that will react with fructose 6-phosphate to form your fructose 1,6-biphosphate. This process is called, brought about by an enzyme called as your phosphofructokinase. Am I clear to you? So this is the first step. Now this I will little explain it even more. I'll start up with this. Please see over here children. The fermentation and respiration are the two major strategies which I already told you. Am I clear to you? So usually the glycolytic pathway is a major metabolic pathway for microbial fermentation which involves a catabolism of glucose into pyruvate. So ಏನಾಗ್ತಿದೆ? Glucose is getting converted into what? Pyruvate. Okay. Now of course it is also called as your EMP pathway because the persons who actually discovered was Emden Mayer of Paranas pathway. Okay. So your glycolysis is also called as ಏನು ಮಕ್ಕಳೇ? Emden Mayer of Paranas pathway, or also called as your EMP pathway. Okay. Now regardless of whether glucose is fermented or respired, it is traveling through the pathway. So thus it is referred as a universal pathway of a glucose catabolism. Okay. So whether it is aerobic or anaerobic, am I clear to you? First it is always your glycolysis. So glycolysis plays a major pathway. Am I clear to you? So who actually discovered is Emden Mayer of Paranas pathway, EMP pathway. Am I clear to you? Moving ahead. Steps involved in the particular glycolysis ಅದನ್ನೇ ನಾನು ನಿಮಗೆ ಇಷ್ಟೊತ್ತು ಮಾತಾಡ್ತಾ ಇದ್ದಿದ್ದು. ಓಕೆ, ಸೊ ಫಸ್ಟ್ ಹಾಫ್ ಅಲ್ಲಿ ಹೇಳಿದೀನಿ, ಆ ಹಾಫ್ ಇನ್ನೊಂದು ಚೂರು ಎಕ್ಸ್ಟೆಂಡ್ ಮಾಡಿ ಮತ್ತೆ I'll go back to the same particular photo. Okay children, now listen up. What is it? In glycolysis, a molecule of glucose. ಮಕ್ಕಳೇ, what do you mean by a molecule of glucose? ಮಕ್ಕಳೇ, a molecule ಅಂದ್ರೆ one molecule of glucose ಅಂತ ಆಯ್ತೇನ್ರಾ? So is degraded in a series of enzymes catalyzed reaction to yield two molecules of the carbon compound pyruvate. Okay, ಏನಂತ ಹೇಳ್ತಾ ಇದ್ದಾರೆ? one the molecule, how many pyruvate it is giving rise to? 2 molecules of your pyruvate. Am I clear to you? So moving ahead, the fermentation of the glucose through a glycolytic pathway can be divided into 2 stages. So glycolysis can be divided into 2 stages. Okay, each requiring several independent enzymatic reactions. ಇವಾಗ ನೀವು ನೋಡಿದ್ರಿ ಫಸ್ಟ್ ಪ್ರಿಪರೇಟರಿ ಫೇಸ್ ಅಲ್ಲಿ ನಾನು ತೋರಿಸಿದಾಗ ನಿಮಗೆ hexokinase ಇತ್ತು, children. So you had all the different enzymes present. Am I clear to you? So children, whenever the reaction is happening, each reaction is having one sweetheart of enzyme. Am I clear to you? Nee nande nan ninge andange. Am I clear to you makle? So each of them are having only one particular enzyme present in them. Am I clear? Okay, moving ahead. Phase 1 comprises of preparatory. So usually glycolysis alli 2 phases ide. Phase 1 and phase 2. Am I clear to you? So comprises of what? Preparatory reaction. These are not redox reaction and do not release energy, but instead form a key intermediate of the pathway. Okay na? So preparatory phase alli nimge yavade reetiyagi energy illa. In fact, ATP is being utilized. Am I clear to you? So there is a consumption of the ATP. Am I clear to you? So there is a consumption of an ATP whichever is happening over there. Am I clear to you, children? So in stage 2, there is a redox reaction occurring where energy is conserved. Okay, so illenagtide stage 2 alli energy is conserved and 2 molecules of pyruvate are formed. So stage 1 and stage 2. Okay na? Stage 1 alli enagtide energy utilize agtide andre utilize agtide, it is consuming. Okay, whereas stage 2 alli enagtide, it is actually consumed and giving rise to the molecules called as pyruvate. So single molecule of the glucose is giving rise to how many molecules of pyruvate? It is giving rise to your 2 molecules of the pyruvate. Am I clear to you? Now let's start up with the phase 1. Okay na? ನಾನು ವಾಪಸ್ ಬರ್ತಿದ್ದೀನಿ, ಅದರಲ್ಲೇನೆ ಇಲ್ಲೇ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ತಾ ಇದೀನಿ, ಕೇಳಿಸ್ಕೊಳ್ಳಿ. Phase 1 is your energy investment phase, also called as preparatory phase. Okay, now first 5 reactions of the pathway. ಇವಾಗ ನಿಮ್ಗೆ ಇಷ್ಟೊತ್ತು ನಾನು ಅಷ್ಟಕ್ಕೆ ನಿಲ್ಸಿದ್ನಲ್ಲ ಮಕ್ಕಳೇ, ಸೊ ಅಲ್ಲಿವರೆಗೂ ನಿಮಗೆ ಫಸ್ಟ್ 5 ರಿಯಾಕ್ಷನ್ ಪಾಥ್ವೇ ಅಂತ ಹೇಳ್ತೀವಿ. ಎಲ್ಲಿವರೆಗೂ? ಒಂದು ನಿಮಿಷ ರೀ, ಇಲ್ಲಿವರೆಗೂ. Am I clear to you? So ಇಲ್ಲಿವರೆಗೂ ನಿಮ್ಗೆ ಏನು ಫಸ್ಟ್ 5 ಪಾಥ್ವೇ ಅಂತ ಹೇಳ್ತೀವಿ ನಾವು. Am I clear to you? Preparatory phase. Okay. See here, children, where it converts one glucose molecule to 2 glyceraldehyde 3-phosphate. Okay, so ಏನಾಗ್ತಿದೆ, glucose molecule is getting converted into 2 glyceraldehyde 3-phosphate, 2 molecules of glyceraldehyde 3-phosphate, where it consumes 2 ATP. ಎಷ್ಟು ATP consume ಮಾಡ್ತಿದೆ? 2 ATP. It includes rate limiting step of conversion of fructose 6-phosphate to fructose 1,6-bisphosphate as catalyzed by your phosphofructokinase. Am I clear to you? ಏನಂದ್ರು ಮ್ಯಾಮು? ಕೇಳಿಸ್ಕೊಳ್ಳಿ. ಇಲ್ಲಿ ನೋಡಿ ಮಕ್ಕಳು, you have glucose. Yes, okay, so this is your preparatory phase. Okay, so glucose is getting converted into what? Glucose 6-phosphate. Am I clear to you? ಇದು ನಿನ್ನ ಫಸ್ಟ್ ಸ್ಟೆಪ್ ಮಕ್ಕಳೇ. By the enzyme ಯಾವುದು? Hexokinase. Am I clear to you? ಯಾರು ಇರೋದು ಇಲ್ಲಿ ಮಕ್ಕಳೇ? Hexokinase. Am I clear to you? So what did I say, children? In the particular preparatory phase ಏನಾಗ್ತಿದೆ, you have your glucose present, where glucose is getting converted into glucose 6-phosphate in the presence of an enzyme hexokinase, where your ATP is getting converted into ADP. ಅಂದ್ರೆ ATP ಏನಾಗ್ತಿದೆ? ಕನ್ಸ್ಯೂಮ್ ಆಗ್ತಿದೆ. ಸೊ ಇಲ್ಲಿ ಎಷ್ಟು ಫಾಸ್ಫೇಟ್ ಇತ್ತು ಮಕ್ಕಳೇ? ಯಾಕಂದ್ರೆ you had 3 phosphate ಅಲ್ವೇನ್ರಾ ಮಕ್ಕಳೇ? ಸೊ ಇಲ್ಲಿ ಏನಾಯ್ತು? ಡೈಫಾಸ್ಫೇಟ್. ಅಂದ್ರೆ ಎಷ್ಟಾಯ್ತು? So it got converted into 2 phosphate. So ಇನ್ನೊಂದು ಫಾಸ್ಫೇಟ್ ಎಲ್ಲಿ ಹೋಯಿತು? It got liberated. So this phosphate whatever is produced was reacted with what? Glucose to form what? Glucose 6-phosphate. Am I clear to you? So ಒಂದು ಹೋಗಿರೋ ಫಾಸ್ಫೇಟ್ ಗ್ಲೂಕೋಸ್ ಜೊತೆ ರಿಯಾಕ್ಟ್ ಆಗಿ ಗ್ಲೂಕೋಸ್ 6-phosphate ನ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದೆ. ಓಕೆ ನಾ ಮಕ್ಕಳೇ? Now Second one, glucose 6-phosphate, so present, is getting isomerized. Glucose and fructose ಏನು ಮಕ್ಕಳೇ ಇವೆಲ್ಲ ನಿಮ್ಮ isomers ಅಲ್ವೇನ್ರೋ? Okay, so your glucose 6-phosphate now getting isomerized into what? Fructose 6-phosphate. So in the presence of enzyme ಏನು? Phosphohexoisomerase. ಏನು ಮಕ್ಕಳೇ? Phosphohexoisomerase. Once again I am telling you, your glucose so present. Okay, so the glucose will react with what ಮಕ್ಕಳೇ? Will react with one of the phosphate. ಹೇಗೆ ಮ್ಯಾಮ್? ATP consume ಆಗಿ ಏನು ಫಾರ್ಮ್ ಮಾಡ್ತು? ADP. ಅಲ್ಲಿಗೆ ಒಂದು ಫಾಸ್ಫೇಟ್ ಲಿಬರೇಟ್ ಆಯ್ತು. ಸೊ ಈ ಫಾಸ್ಫೇಟ್ ಗ್ಲೂಕೋಸ್ ಜೊತೆ ರಿಯಾಕ್ಟ್ ಆಗಿದ ತಕ್ಷಣ ಏನು ಫಾರ್ಮ್ ಮಾಡ್ತು? ಗ್ಲೂಕೋಸ್ 6-phosphate ಫಾರ್ಮ್ ಮಾಡ್ತು. Now, glucose 6-phosphate is getting isomerized in the presence of what? Phosphohexose isomerase. Am I clear to you? So, phosphohexose isomerase ಅನ್ನೋ ಒಂದು enzyme ಜೊತೆಗೆ when it reacted, it forms what makla? It forms your fructose 6-phosphate. Am I clear to you? So, for the isomerization people, you require what enzyme makla? You require your phosphohexose isomerase. Okay, moving ahead. When your fructose 6-phosphate, okay, now in the presence of an enzyme phosphofructokinase, okay, what will happen? It will get converted into fructose 1,6-bisphosphate. ಇಲ್ಲಿ ನೋಡಿ 6th ಕಾರ್ಬನ್ ಆಟಮ್ ಅಲ್ಲಿ ನಿಮಗೆ ಫಾಸ್ಫೇಟ್ ಇದೆ, ಕರೆಕ್ಟ್ ಏನ್ರಾ? ಸೊ ಇಲ್ಲಿ ಮತ್ತೆ 1 ಅಲ್ಲೂ ಇದೆ 6 ಅಲ್ಲೂ ಇದೆ. So how did that another one came? Simple again. Another ATP is getting consumed to form ADP. Again one phosphate is liberated. This will react with fructose 6-phosphate. Now this phosphate is getting attached to which carbon? It is getting attached to your first carbon. Okay, so that is how you have got your fructose 1,6-biphosphate. ಅರ್ಥ ಆಗ್ತಿದ್ಯಾ? ಗ್ಲೂಕೋಸ್ 6-phosphate ಹೆಂಗ್ ಬಂತು ಮ್ಯಾಮ್ ಫಾಸ್ಫೇಟ್ ಅಲ್ಲಿ? ATP is got converting into ADP, one phosphate liberate ಆಯ್ತು, ಅದೇ phosphate got attached to your glucose. Now darling, that particular in the presence of an enzyme hexokinase, what did it form? Glucose 6-phosphate. Now glucose 6-phosphate in the presence of phosphohexose isomerase, what did it form? It went isomerization to form fructose 6-phosphate. Now again fructose 6-phosphate is forming fructose 1,6-phosphate. How? ATP is getting again converted into ADP, one phosphate is liberated which is reacted with fructose 6-phosphate in the presence of an enzyme phosphofructokinase. So what will be formed? Fructose 1,6-biphosphate. Now darlings, your fructose 1,6-biphosphate so present in the presence of the enzyme called as aldolases. Again it will undergo your particular process to give rise to two products over here. ಯಾವ್ಯಾವುದು? Glyceraldehyde 3-phosphate and dihydroxyacetone 3-phosphate. ನಿಮ್ಗೆ ಈ ಲಾಸ್ಟ್ ಸ್ಟೆಪ್ ಮ್ಯಾಮ್ ಒಂಚೂರು ಅರ್ಥ ಆಗ್ಲಿಲ್ಲ, ಅದು ಯಾಕಂದ್ರೆ ಪ್ಲಸ್ ತರ ಇದೆ ಅಂದ್ರೆ ನೀವು ಯೂಶಲಿ ಈ ರೀತಿ ಬರೀತಿದ್ರಿ ನೋಡಿ, ಈ ರೀತಿ ಬರೀತಿದ್ರಿ ಅಲ್ವೇನ್ರಾ? ಸೊ fructose 1,6-biphosphate, okay, now will undergo in the presence of an aldolases will form glyceraldehyde 3-phosphate and dehydroxyacetone phosphate. Am I clear to you? So idu ಏನು ಮಕ್ಕಳೇ, both of them are they are the reversible reaction. So this can get converted into this and this can get converted into this. Am I clear to you? So the particular two things are also what makle? They are the what to say children? They are actually getting converted into each other. Okay na? So fructose 1,6-bisphosphate gave rise to glyceraldehyde 3-phosphate, dehydroxyacetone phosphate. Am I clear to you? So these two products are formed. ಇವಾಗ ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅಂತ ಕ್ಲೀನ್ ಆಗಿ ಕೇಳಿಸ್ಕೊಳ್ಳಿ. ಇವಾಗ ಇಲ್ಲಿದೆ ಅಲ್ವಾ ಈ ಪರ್ಟಿಕ್ಯುಲರ್ glyceraldehyde 3-phosphate, darling, no doubt it will give rise to your pyruvate. Okay na? But what is the fate of DHAP? DHAP will get converted into your particular glyceraldehyde 3-phosphate. ಅರ್ಥ ಆಗ್ತಿದ್ಯಾ? ಸೊ DHAP ಏನು ಕನ್ವರ್ಟ್ ಆಗ್ತಿದೆ ವಾಪಸ್ಸು glyceraldehyde 3-phosphate ಗೆ ಕನ್ವರ್ಟ್ ಆಗ್ತಾ ಇದೆ. Am I clear to you? So this is what happens. So ಯಾವಾಗ DHAP ಸಹ when the DHAP is also getting converted into your glyceraldehyde 3-phosphate, children, from this you are getting one pyruvate, correct? So when this thing gets converted into glyceraldehyde 3-phosphate, don't you think you will get another pyruvate? So that is how you are getting two pyruvates. Am I clear to you? So this is how you are getting 2 pyruvates. One molecule in the, how did you get to 2 pyruvate molecules? Because from glyceraldehyde 3-phosphate, you are already getting pyruvate. So from another one also is getting converted, and that is also forming a pyruvate. Understood, Makla? Yes? This is how you will be having a preparatory phase. So up till so much of phases, you have your particular, what to say, preparatory phase. Next one. The same thing till now, whatever I have explained, the same thing I have explained it in particular writing students. So moving ahead. Phase 2. Okay. Now this is a payoff phase. Darling, here where your energy is being produced. So there the energy had been consumed. So here it is getting paid off for whatever it has taken in. Am I clear to you? So in the payoff phase, what is that? This involved a reaction. The first redox reaction is the oxidation of glyceraldehyde 3-phosphate to 1,3-biphosphoglyceric acid. Am I clear to you? So what is the first step, children? ಏನಂತ ಹೇಳ್ತಿದ್ದಾರೆ ನೋಡಿ, ಇದು first step ನೋಡ್ಕೊಳ್ರೋ. ಇವಾಗ ಪೇ ಆಫ್ ಅಲ್ಲಿ ಏನಾಗ್ತಿದೆ? The first redox reaction, I'll just write it for you. See, the first redox reaction is the oxidation of what, makle? Glyceraldehyde 3-phosphate. Am I clear to you? Glyceraldehyde 3-phosphate to form what? To form your particularly 1,3-biphosphoglyceric acid. Am I clear to you? What is it forming, makla? 1,3-biphosphoglyceric acid. Am I clear to you? So this is the product. Am I clear? What is it? 1,3-biphosphoglyceric acid. Am I clear to you? By what enzyme? So enzyme is different here. What is the enzyme, makla? The enzyme is nothing but your glyceraldehyde 3-phosphate dehydrogenase. Am I clear to you? So what is happening, children? So the first redox reaction is the oxidation of the glyceraldehyde 3-phosphate to form what? To form 1,3-biphosphoglyceric acid by what? By glyceraldehyde 3-phosphate dehydrogenase. And here it uses the cofactor NADH+. Okay, na? So that's your first step. Next. Moving on to the next particular step, makla. Where? Second step here, with the 1,3-biphosphoglyceric acid, okay, is getting converted into what? It is getting converted into 3-phosphoglyceric acid. ಏನು ಕನ್ವರ್ಟ್ ಆಯ್ತು ಇವಾಗ ಮಕ್ಕಳೇ ನೋಡ್ರೋ ಇಲ್ಲಿ you had your phosphoglyceric acid, yes or no? Okay, you had your phosphoglyceric acid over here. So the same thing we are written over here. So phosphoglyceric acid, now it is getting converted into what? 3-phosphoglyceric acid. What is it getting converted into, makla? 3-phosphoglyceric acid. By what? By a particular enzyme. Okay, by the enzyme phosphoglycerate kinase. Okay, so this reaction alli enagutte makle, you are particularly very important. Okay, very importantly, you have your 2 ATP produced. What is produced, makle? 2 ATP is produced. Am I clear to you? As per the glucose molecules. Am I clear to you? Enagtide 1,3-5-phosphoglyceric acid is getting converted into 3-phosphoglyceric acid by the enzyme phosphoglycerate, where 2 ATPs be produced. Am I clear? Moving ahead. Now, the reversible conversion of yadu 3-phosphoglyceric acid to 2-phosphoglyceric acid is taking place. Am I clear to? What is it getting converted to now? It is getting converted into 2-phosphoglyceric acid. What is it getting converted to, makla? It is getting converted to your 2-phosphoglyceric acid. The enzyme which is involved, yadu makla? Phosphoglycerate mutase. Okay, moving ahead, the next one, reversible conversion of 2-phosphoglycerate to what? Finally, it is getting converted into what, makla? It is getting converted into your phosphoenol pyruvate. What is it getting converted into? Phosphoenol pyruvate. By the enzyme what, makla? By the particular enzyme, your enolase. Okay. Now, the last step, where it is an irreversible reaction. Andre reverse agakagala, where the conversion of a particular PP. What is a PP? Phosphoenol pyruvate. Okay, finally it is getting converted into what, Makle? It is getting converted into your particular pyruvate. What is it getting converted into? Pyruvate. Am I clear to you? Where? What is the enzyme involved? Pyruvate kinase. What is the enzyme involved, Makle? It is nothing but your pyruvate kinase. Okay, there is a net gain of a 2 ATP per glucose molecule in this reaction. Hey, come on, that I will tell you. Ilori, these two are reversible. Yes. In the presence of a particular triose phosphate dehydrogenase, what happened? 1,3-biphosphoglycerate was formed, where your 2 NAD got reduced to 2 NADH plus 2H plus got produced. Again, 1,3-5-phosphoglycerate got converted into what? 3-phosphoglycerate, where your ADP is getting converted into what? ATP. So your particularly one phosphate is being added and ATP energy is being released. Am I clear to you? Other ರೀತಿ, in the presence of your glycoromutase, what happens? 2 phosphoglycerate is formed. Finally, by the enolase, where 2 phosphoglycerate, there is a dehydration happening where your PP is being formed. Finally, what is happening? In the presence of your pyruvate kinase, pyruvate is formed where your 2 ADP, again there is a production of your 2 ATP. Am I clear to you? So basically there are particularly 2 to 4 ATPs which is being produced. Am I clear to you? Moving ahead. Nodi, during the stage 1 and stage 2 of glycolysis, 2 ATP molecule are consumed initially, okay, and how many ATPs are produced? 4 ATP molecules are synthesized, okay. Thus the net energy yield in glycolysis is 2 molecules of ATP per molecule of the glucose fermented. See children, this is what, from glucose totally how many ATPs have been produced? 4 ATPs have been produced. Am I clear to you? So NADH is to 2 NADH. Am I clear to you? So always whenever you have your NADH multiply with 2, so totally you will have 4 ATP produced. Again you have 2 NADH, so you have a particular 6 ATP produced. Am I clear to you? And finally you will have a particular Krebs cycle in the you will have so much of, I will teach about this balance sheet little later. Okay, so that it will be easy for you. So totally there will be usually a 36 particular thing. However, maximal ATP yield of oxidation of glucose is 36 to 38. Am I clear to you? I will tell you about this little later. Keep this in your mind. I'll speak about it little later. Now, significance of a glycolysis pathway. Now, what is the significance of glycolysis? The glycolytic pathway is usually employed by all tissues for breakdown of glucose to provide energy in the form of ATP. Okay, so it provides energy. Okay, important pathway for the production of energy, especially under anaerobic respiration. So anaerobic respiration alli nimge important pathway energy vehicle so that energy whichever is produced in this is usually utilized in anaerobic. It is a crucial for generation of energy in cells without mitochondria. Okay, so yavyavdu mitochondria alva adrella nimge energy produce agirade use agadu. It forms the products that are intermediate for other metabolic processes. So glycolysis interface with glycogen metabolism. Okay, so the pentose phosphate pathway, the formation of amino sugars and triglycerides. So there is another pathway called as pentose phosphate where there also the intermediates of these things are used up. Now children, this is a overall thing. I don't go in detail with that, but you just see the first this one and the last column. Okay, so hexokinase usually they are phosphoryl transfer. Okay, then phosphoglucoisomerase they usually help in isomerization. Phosphofructokinase they usually help in your particular phosphoryl transfer. Aldolases they help in your aldo cleavage. Am I clear to you? Next, phosphotriose isomerase they usually help in your isomerization. Next, glyceraldehyde 3-phosphate dehydrogenase helps in your phosphorylation coupled to oxidation. Phosphoglycerate kinase they usually help in your phosphoryl transfer. Phosphoglycerate mutase help in your phosphoryl shift. Okay. Next, enolase helps in your dehydration. Pyruvate kinase usually helps in your phosphoryl transfer. Okay, Makla. So these are the important enzymes. What are the functions of them? Are the main things. Okay. So later ahead you can see through the whole chart also, not a problem, but those two are important. Moving ahead, ATP formation. Yeah, let's start up. 1,3-diphosphoglyceric acid ಅಲ್ಲಿ ನಿಮಗೆ product produce ಆಗೋದು 2. Okay na? So in terms of ATP also there are 2 ATPs. Am I clear? So totally nimge 1,3-diphosphoglyceric acid. Okay, to form a 3-phosphoglyceric acid and phosphopyruvic acid to pyruvic acid. Okay, first in the second one 1,3-diphosphoglyceric acid in the 3-phosphoglyceric acid 2 moles of molecules. So total agi nimge ಎರಡರಿಂದ ಎಷ್ಟು ಬಂತು 2 ATP 2 ATP. So total agi ಎಷ್ಟು ಬಂದಿದೆ 4 ATP. ಅದೇ ರೀತಿ ATP formation of oxidative ETC ಅಲ್ಲಿ ನಿಮಗೆ ಟೋಟಲ್ ಆಗಿ NADH ಯಾವಾಗ ಎಷ್ಟಾಗಿದೆ 2. Always remember 2 will NADH ATP produce ಮಾಡಬೇಕಂದ್ರೆ it will be multiplied with 3, so that you will get your 6. Am I clear to you? Next, in this particular thing, so 6 plus 4 totally you got how many ATP? 10 ATP. Am I clear to you? So ATP consumed in glycolysis. So glucose molecule to glucose 6-phosphate to fructose 6-phosphate to fructose 1,6-phosphate. ಅರ್ಥ ಆಗಿಲ್ಲ? Glucose to glucose 1,6-phosphate ಅಲ್ಲಿ 1 ATP ಹೋಯ್ತು. Fructose 6-phosphate to fructose 1,6-phosphate ಅಲ್ಲಿ ಮತ್ತೆ 1 ATP consume ಆಯ್ತು. ಅರ್ಥ ಆಗ್ತಿದ್ಯಾ? So total ಆಗಿ how many ATP was consumed? 2 ATP was consumed. So if you make a total net charge, totally how many ATP was produced? 10. So 10 minus 2, totally you have got 8 ATP in glycolysis. Okay. The same thing, children. The total output what they have written. Next, we will start up with your Krebs cycle, which is also called as a citric acid cycle. It is also called as TCA cycle because it is a tricarboxylic acid cycle. So it was a scientist named Krebs who actually discovered the Krebs cycle, hence the name Krebs cycle. Krebs cycle is contained, or it happens within the mitochondria. Okay, so within the, where in mitochondria? In the mitochondrial matrix. The reactions of the Krebs cycle adds electron and protons to number of electron carriers, which are then used by electron transport chain to produce the ATP. Okay. Now, the TCA cycle takes place over 8 different steps. So then ನಾನು ಇವಾಗ 1 by 1 steps ಹೇಳ್ಕೊಂಡು ಹೋಗಿರ್ತೀನಿ. ಓಕೆನಾ? ಅದಾದ್ಮೇಲೆ ಮತ್ತೆ ನೀವು ಓವರಾಲ್ ನೋಡಿರಂತೆ. Step 1, the first acetyl-CoA joins with oxaloacetate. Now, point is over here. Ma'am, acetyl-CoA ellinda bantu from the pyruvate. Okay. To form the citrate. Now, second step, the citrate is then converted into isocitrate. Next one, isocitrate is then oxidized to alpha ketoglutarate, which is a 5-carbon atom, which results in the release of a carbon dioxide of 1 NADH molecule is also formed in this step. Now, the enzyme responsible for this is isocitrate dehydrogenase. Step 4, alpha ketoglutarate is oxidized to 4-carbon molecule, which picks up a coenzyme A, forming your succinyl-CoA. This conversion is also forms the NADH molecule. Succinyl-CoA is then converted into your succinate and 1 GTP molecule is produced. Succinate is converted into fumarate and a molecule of FADH2 is also produced. Okay, so fumarate is converted into your malate and malate is then converted back to your oxaloacetate and NADH is also produced over here. Now, before this, we will go to a cycle immediately and then we will go back to that. Okay, so see over here, children, the pyruvate. Okay, so pyruvate, in the presence of your pyruvate dehydrogenase, where your coenzyme A will get converted into carbon dioxide and NADH to form. So your CoA will react with pyruvate to form your acetyl-CoA. Okay. Now what happens? Now children, your acetyl-CoA reacts with your oxaloacetate to form, first one is your citrate, the first compound. Am I clear to you? That is your citric acid cycle. So now citrate will, in the presence, and one, by the way, citrate is produced or synthesized by an enzyme called as citrate synthetase. Now this reaction requires your addition of the water. Am I clear to you? What does it require? It requires your addition of the water. Now citrate will get converted into isocitrate by the enzyme called as aconitase. Okay. Now, isocitrate will get converted into alpha ketoglutarate in the presence of an enzyme called as isocitrate dehydrogenase, where NAD is getting reduced to your NADH. Next, alpha ketoglutarate is getting converted into your succinyl CoA by the presence of an enzyme alpha ketoglutarate dehydrogenase. Again over here, your NAD is getting reduced to NADH plus carbon dioxide is produced. Also, there is an addition of coenzyme A. Therefore, when all these things are added, okay, the 5-carbon atom, that's your alpha ketoglutarate, will get converted into your succinyl CoA. Now, succinyl CoA will get converted into your succinate by the presence of an enzyme succinyl CoA synthetase, where there is a removal of the coenzyme A, where your GDP will get converted into your GTP. Am I clear? Next, your succinate in the presence of your succinate dehydrogenase will get converted into fumarate, where again your FAD will also be there. Ili GDP jote FAD nu barutte. Am I clear to you? Next, your fumarate will undergo your hydration, means to say addition of the water in the presence of a fumarase to form your malate. Now, malate will again reduce your NAD to NADH2 in the presence of your malate dehydrogenase to form your oxaloacetic acid, nothing but your oxaloacetate. Am I clear to you? So this is the entire cycle of what makla? It is the entire cycle of a particularly the Krebs cycle, nothing but your citric acid cycle. The same thing, children, the enzyme and the last box. You see this box and you see the first box. Am I clear to you? That is this box. Okay, so let's start up over here. Now, what does each and everything are doing? Citrate synthetase is helping in your condensation. Iconitase, what is it doing? It is helping in a isomerization. Isocitrate, what is it doing? Isocitrate dehydrogenase, helping in your oxidative decarboxylation. Alpha ketoglutarate dehydrogenase complex, what is it doing? Oxidative decarboxylation. Succinyl CoA synthetase, what is it doing? Substrate level phosphorylation. Succinate dehydrogenase, it is your helping in oxidation. Fumarase, it is helping in hydration. Malate, it is helping in your oxidation. Am I clear to you? So these are the important types of your particular enzymes. Now let's start up. When succinyl CoA getting reduced to your succinic acid, 2 GTP is produced. When 2 GTP is getting converted into ATP, you always multiply it with same number of one molecule. So 2 into 1 is nothing but a 2 ATP. So total H produce alkali, 2 ATP produce again. Next up, pyruvic acid to acetyl CoA, where you have 2 NADH. Now avagale helide NADH will always be multiplied with 3, so you will get 6 ATP. Isocitric acid, oxalosuccinic acid convert adaga, again you have this, so 3, 6 ATP. Alpha ketoglutaric into succinyl CoA, so 3, 6 ATP. Okay, so succinic acid into fumaric, 2 FAD. Again children, you will multiply with 2. So 2 into 2 is 4. Malic acid to oxaloacetic acid, NADH, so it is 3. So 3 2s are 6. So totally how many? So if at all you add up, it will come up to your 28 ATPs. So 28 plus 2 ATP over here. Okay, so how much you will get? 30 ATP. ಇವಾಗ ಲೆಕ್ಕ ಹಾಕೊಳ್ಳಿ. ಇಲ್ಲಿ 30 ಅಲ್ಲಿ ಎಷ್ಟು ಬಂತು 8, so 38. Am I clear to you? So this is the entire process. See the same thing. Okay, so ATP in glycolysis. So what happens children over here? 8 ATP plus 30 ATP. So totally how much you got? 38 ATP. Now, whereas in your particular oxidative phosphorylation, what will happen? 6 ATP plus 28 ATP ಬರುತ್ತವಾಗ ಅವಾಗ ನಿಮಗೆ ಏನಾಗುತ್ತೆ 34 ATP ಬರುತ್ತೆ. Now we are going to your electron transport chain. Okay. Now coming to your electron transport chain, what is happening? Children, here NADH is getting converted into NAD. Now you know oxidation and reduction process, if I'm not wrong. Yes or no? So oxidation reduction alli enagutte? Oxidation means always remember it is giving the particular electron. Reduction means what? It is always you're receiving the electrons. Am I clear to you? So whenever a particular NAD is giving the particular electron, okay, so they are getting reduced. Am I clear to you? So whenever NAD is to NADH, then again you see the same thing follows. So the electron from NAD is passing on to your cytochrome B. From cytochrome B, it is getting cytochrome C. So again, oxidation and reduction. So from this, it is going to cytochrome A. From this, it is going to cytochrome A3. Here, it is not there. Okay. And finally, what happens? Your particularly oxygen and hydrogen is produced in the presence of your particular water. There you have your ATP synthetase, also enzyme present. Okay. So this is what I have written. So hope it will be easy for you. NADH and FADH is formed in glycolysis TCA cycle. Okay. So ATP is generated as a result of what? Energy producing electrons from NADH2 and FADH2 are passed to your molecule of oxygen by a series of electrons, collectively known as what, children? Electron transport chain. Okay. So this chain is completely called as ATP is generated as a result of energy produced when electrons from NADH and FADH are passed to molecular oxygen by a series of carriers. Andre oxygen final electrons receive agtade by who? By all these electron carriers. So this is nothing but your electron transport chain. Okay. Now electron transport chain is also called as cytochrome oxidase system, also called as your respiratory chain. Okay. So remember, electron transport chain is also called as cytochrome oxidase system, also called as a respiratory chain. The component of the chain include FMN. Okay. Then it also requires FES, that is your ion centers, sulfur centers, and coenzymes Q and your particular series of cytochromes, that is your B, C1, C and A and A3. Am I clear to you? So A and A3. Okay. So the energy derived from the transfer of electrons through the electron transport chain is used to pump protons across the inner mitochondrial membrane from the matrix to cytosolic side. So usually they are again happening in your particular mitochondria. Now children, let's start up. See, it's on the second point. Okay. The oxidation of the 1 mole of NADH generates approximately 2.5 molecules of ATP. Whereas oxidation of 1 molecule of FADH generates approximately 1.5 moles of ATP. Because of the energy generated by transfer of electrons to the electron transport chain to oxygen is usually used in the production of ATP. So, ಈ energies ಎಲ್ಲ use ಆಗ್ತಾರೆ electron transfer ಆಗ್ತದೆ ಏನಕ್ಕೆ ATP produce ಮಾಡೋದಕ್ಕೆ. So, the overall process is known as your oxidative phosphorylation. What is it usually called as makla? Oxidative phosphorylation. Now location of ETC ಎಲ್ಲಿ ಮಕ್ಕಳೇ? Obviously it is located in cytoplasmic membrane of the bacteria, but in case of eukaryote it is located in the membrane of mitochondria. Okay. So these are the ones. Okay. ಇಲ್ಲಿ ನೋಡಿ, where your complex 1 you have, complex 2, complex 3, okay, complex 4 and complex 5. So complex 5 is the last one. Am I clear to you? So this is how usually your chain happens. Am I clear to you? Now, complex 1, it is NADH dehydrogenate. It contains FMN which accepts 2 electrons and hydrogen from 2 NADH, okay, to become reduced to what? To form your FMNH2, okay. So what will happen? It will get reduced to FMNH2. Also contains ion atom which assists the transfer of the electrons and your protons. Now, complex 2, what does it contain? Succinate dehydrogenate. It contains ion and succinate which oxidizes FAD to FADH2. Okay. Now, third one is your coenzyme Q. It accepts electron from FMNH2 and FADH2 and transfers electrons to complex 3. So, complex 3 contains cytochrome B. It contains heme group in which Fe3 plus accepts the electron from coenzyme Q to become Fe2 plus. So, transfers the electrons to next one what? It transferred to cytochrome C. So, cytochrome C ಏನು ಮಾಡುತ್ತೆ? It contains the heme group in which Fe3 plus accepts the electron from complex 3 to become again Fe2 plus. So, transfers the electrons to the complex 4. Now, Complex 4 is nothing but your cytochrome A. Now here what is happening? It contains the heme group in which Fe3 plus accepts electrons from cytochrome C to become Fe2 plus. So transfers the electrons to oxygen which combine with hydrogen to form what? ATP. So finally what happens? Complex 5. It contains a proton channel that follows the protons to across the matrix using a proton gradient energy to form your ATP. ಅರ್ಥ ಆಯ್ತೇನೋ? ಮತ್ತೆ ಹೇಳ್ತೀನಿ ಕೇಳಿಸ್ಕೊಳ್ಳಿ. ಕಾಂಪ್ಲೆಕ್ಸ್ 1 ಏನಾಗಿರುತ್ತೆ? NADH dehydrogenase ಇರುತ್ತೆ. It contains what? FMN, which accepts what? FMN accepts what? 2 electrons and the proton to form what? To form a particular 2 NADH. Okay, sorry, 2 electrons from what? Not to form, from what? In electrons ಎಲ್ಲಿಂದ accept ಮಾಡ್ಕೊಂತು? NADH ಇಂದ ಬರ್ತಾ ಇದೆ ಇವಾಗ. ಎಲೆಕ್ಟ್ರಾನ್ಸ್ ಬರ್ತಿರೋದು NADH ಇಂದ. ಸೊ ಬಂದಿದ್ದ ಎಲೆಕ್ಟ್ರಾನ್ಸ್ ಯಾರಿಗೆ ಆಕ್ಸೆಪ್ಟ್ ಆಗಿದೆ? FMN ಗೆ ಆಕ್ಸೆಪ್ಟ್ ಆಗಿದೆ. So once FMN accepts the electron, it will get reduced to what? FMNH2. Okay. Now, which assists in the transfer of the particular electron and the proton to coenzyme Q. Now, complex 2. What it contains? Succinate dehydrogenase. Ilenagutte, it contains ion and succinate, which oxidizes FAD to form what? FADH2. Am I clear to you? So ಅಲ್ಲೇನಾಯ್ತು, ಅಲ್ಲಿ FMN ಇಂದ FMNH2 ಇಲ್ಲಿ ವಾಪಸ್ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗಿದೆ ಅದಕ್ಕೆ FAD ಗೆ. Now FAD got converted into what? FADH2. Okay, it oxidized. Next, coenzyme Q. It accepts electrons from FMNH2 and FADH2. ಎರಡರಿಂದನೂ ಬಂದಿದೆ ಅಲ್ವಾ, ಎರಡರಿಂದನೂ accept ಮಾಡಿ it transfers the electrons to what? Complex 3. Complex 3, what does it contains? It contains a cytochrome B. So it contains heme group in which Fe3 plus accepts the electrons from coenzyme Q to become Fe2 plus. So transfers the electron to cytochrome C. So cytochrome C, what is happening? It contains a heme group in which Fe3 plus accepts the electron from complex 3 to become Fe2 plus. So transfer of electrons to your complex 4. Afterwards, LE transfer to complex 4. Next, complex 4 is cytochrome A. So it contains the heme group in which Fe3 plus accepts the electron from what? It is accepting the electrons from your cytochrome C to become what, McLean? To become a particular Fe2 plus and transfers the electrons to oxygen. So finally, electrons is going to what? It is going to your oxygen, in which it is combining with what? Hydrogen to form what? Water. Finally, the protons are passing through your ATP synthetase to liberate what? ATP. It is nothing but your ATP synthetase is your fifth complex. Now that is what I explained you. This is in detail. So this is your ATP synthetase. Am I clear too? This is your ATP synthetase where your ATP is produced. See, the proton gradient which is created will pass through your ATP synthetase where your ADP is getting converted into ATP by addition of your inorganic phosphate. Am I clear to you? This is also called as what, McLean? Phosphorylation. Now see, from glycolysis 8, I have already. Okay, acetylation of pyruvic acid also 6. Krebs cycle 24. So total net change is how much? 38. So this is your 38. So how did 36 come? Sometimes mitochondria which are produced outside the mitochondria can give rise to only 2 ATPs under physiological condition. Aವಾಗ ನಿಮಗೆ ಬರೀ 36 ಬರುತ್ತೆ. Okay, so this is your balance sheet. See ಮಕ್ಕಳೇ, ಒಂದ್ಸಲಿ ನೋಡ್ಕೊಂಬಿಡಿ. Glycolysis, so 4 to 6 ಬರುತ್ತೆ. Okay, oxidative 2 NAD 6, so Krebs cycle 6 NAD 18, 2 FAD 4, 2 ATP 2. Total is 36 to 38 ATP. Next we are going to your particularly a respiratory quotient. Okay, here makla the respiratory quotient. Now makla, what do you mean by the respiratory quotient? The respiratory quotient or RQ value is a measure of the ratio of carbon dioxide produced to oxygen being consumed by the organism per unit time. ಏನಂತ ಹೇಳ್ತಿದ್ದಾರೆ ಇಲ್ಲಿ ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡ್ ಎಷ್ಟು ಪ್ರೊಡ್ಯೂಸ್ ಆಗಿದೆ to what? To your volume of oxygen consumed. Am I clear to you? So this one you usually keep it in your mind, and usually there is no units for your respiratory quotient. So let's start up. RQ, volume of carbon dioxide produced by volume of oxygen consumed. So the respiratory quotient is valuable measurement as it provides us with the information regarding the nature of substrate being used by the organism for the respiration. So RQ ನಮಗೆ ಏನು ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟ್ ಆಗುತ್ತೆ ಅಂದ್ರೆ if at all an organism is producing an energy, what was the substrate used by it will play a very important role. Okay, so the simplified equation for aerobic respiration of glucose is, so see here how much is there. So again let's calculate how much of the carbon dioxide is being produced. Makla 6 is produced, yes or no? So how much of oxygen is consumed here, makla? 6. So what is the answer, makla? Obviously it will be 1. Am I clear to? So this is what they have shown you over here. 6 carbon dioxide molecule as produced by your 6 oxygen molecules. So therefore the RQ value will be usually how much, makla? It will be usually 1. Now just know this, fatty acids is usually 0.7, protein is 0.9. Okay, so this is what is your RQ value for glucose, fatty acids and your proteins. What is it? 1, 0.7 and 0.9. Now next one is your amphibolic pathway. In makla, next we are going ahead with your amphibolic pathway. Now children, in the year 1961, B. Davis coined a particular term, amphibolic pathway. Now what is it? Any biological pathway which involves both metabolic process, that is your catabolism and anabolism. ಎರಡೂ ಇದ್ರೆ ಕ್ಯಾಟಬಾಲಿಸಮ್ ಇದ್ರೆ ಅನಾಬಾಲಿಸಮ್ ಇದ್ರೆ ಏನಂತ ಕರಿತೀವಿ, ಆಂಫಿಬೋಲಿಕ್ ಪಾಥ್ವೇ ಅಂತ ಕರಿತೀವಿ. Am I clear to? So let's start. Amphibolic pathway, respiration is the breakdown of the complex compounds into what? Simple ones to produce what? Energy. That is nothing but your ATP. ಏನು ಹೇಳಿಲ್ಲ, respiration ಅಂದ್ರೆ you're breaking down of your complex compound. Okay, to what? To the simple molecules. And what is liberated? ATP is being liberated. Hence, the process is called as your catabolic process. So basically, the particular breaking down, and the pathway is termed as your catabolic pathway. Actually, the respiration is the result of both making and breaking. Am I clear to you? So when energy is required, proteins or fatty acids are broken down. Okay, ಇಲ್ಲಿ break ಆಗ್ತಾ ಇದೆ to form what? Acetyl-CoA and further process of respiration. Now this is called as your catabolism. Am I clear to you? So when the body requires fatty acids and proteins, respiratory pathway is stopped and same acetyl-CoA is utilized by the fatty acids and are manufactured by the respiration. Now this process of synthesis and then making is termed as anabolism. Breaking is called as catabolism. Making is called as anabolism. Thus we can say respiration is the sum of process of catabolism and anabolism. Hence the respiratory pathway is known as your amphibolic pathway. Okay, so this is your amphibolic pathway. Okay, now I'll do one thing. First I'll read this, then I'll go ahead with your NCERT list so that it will be easy for you because you already studied that. So before entering into respiration, carbohydrates are the first converted into what? Glucose. And other substrate can also be respired. For example, fats. Okay. Now fats to be broken down into glycerol and fatty acid first. Fatty acids needs to be broken down into acetyl-CoA. And then they can enter into the pathway. Okay, so fats is breaking down into fatty acids and glycerol. Then it is getting converted into acetyl-CoA and then it is entering into your pathway. That's your Kreis. Okay, next. Similarly, glycerol is getting converted into PGAL and then can be entered into your pathway. That is depending on your glucose. Am I clear to you? So glucose glycerol, whatever is present, is converted into what? PGAL, phosphoglycerolic acid, and then what is happening? It entering into your pathway again. In proteins, it is first broken down into amino acids and then it is entering into your pyruvic acid and enter into your respiratory pathway. So this is nothing but your amphibolic pathway. Illu nodi nimge ide kotirodu eno, fats is getting converted into fatty acid, then it is getting converted into acetyl-CoA reaching your Krebs cycle. Okay, next carbohydrate, simple glucose is getting converted into glucose 6-phosphate, fructose 1,6-phosphate, DHAP, glyceraldehyde 3-phosphate. So glyceraldehyde 3-phosphate finally entering into your Krebs cycle by what? Pyruvic acid, acetyl-CoA and Krebs cycle. Whereas proteins, okay, it is getting converted into amino acid. So amino acid is getting converted into pyruvic acid. Finally, it is reaching up to your Krebs cycle. So this is nothing but your amphibolic pathway. Now let's start up with your MCQs. Okay, makla. So now starting up with your MCQs. Respiratory substrates are the organic substances which are dashed during the respiration to liberate energy. I repeat again, respiratory substrates are the organic substances, of course, which are dashed using during the respiration to liberate energy. What are they undergoing? Are they getting oxidized, reduced or synthesized? Makle, whenever there is a respiration or an energy released, always remember, I told you, Makle, what is it? Always we will have a particular oxygen being added. So for your organic food, first particular definition for organic compound, what is happening? Oxidation is happening. When we say oxidation, what is it? Always your particular oxygen being produced. Am I clear to you? So whenever your oxygen is added to your organic substances, what will happen? It will get oxidized but not reduced. Am I clear to you? So what is happening? Oxidized. Okay. So what is the first option? It is nothing but your A. Second one, flow chart. See, Magla, it is easy. Identify what is A, B, C and D. It is very easy. Okay, glucose. Okay, so what is glucose getting converted into? Glucose 6-phosphate. What is glucose 6-phosphate getting converted into? It is getting converted or isomerized on the head of the better word ಏನು ಮಕ್ಕಳೇ? Isomerized. So what is it called as? Fructose 6-phosphate. Am I clear to you? So fructose 6-phosphate is getting converted into what? Fructose 6-biphosphate. Now fructose 6-biphosphate is getting converted into what? It is getting converted into phosphoglyceraldehyde. Okay, now so after that, what is happening? Your phosphoglyceraldehyde, where there is a reduction, your NAD will get reduced to NADH, where it will form what? 1,3-biphosphoglyceric acid. Yes or no, children? So what will be the option? Obviously, it will be a particularly the first option. What is it? A. Third one. Match the columns. Okay, so match the following. Okay, again, let's start up. Okay, what is it? Now, see, children, in the particular third question, what is there? Glycolysis. Okay, what is happening in glycolysis? TCA cycle and ETS. Am I clear to where exactly all these things are taking place? ಯಾವ place ಅಲ್ಲಿ you have your particular thing happening. Am I clear to you? So glycolysis ellagutte? Obviously ellagutte kandro inner mitochondrial membrane, mitochondrial matrix or cytoplasm. Where it will happen, makle? Definitely, if at all you look into very closely, what is the direct answer? It is nothing but your cytoplasm. TCA cycle, where is it happening? Of course, mitochondria, no doubt. But think for a while, where it is happening? Mitochondrial matrix ಅಲ್ಲಿ ಆಗ್ತಿದ್ಯಾ ಅದು particular mitochondrial membrane ಅಲ್ಲಿ ಆಗ್ತಿದ್ಯಾ? Obviously, what is it, children? It is mitochondrial matrix. So TCA cycle is happening where, children? Mitochondrial matrix. So they have given direct answer. Next one, third one, ETS. So ETS ಎಲ್ಲ ಆಗ್ತಿದೆ? Inner mitochondrial membrane. Am I clear to? So what is the option, children? The option is nothing but your B. What is the option? The option is nothing but your B. Next one. Select the wrong statement with respect to your glycolysis. What is it, children? Select the wrong statement with respect to your glycolysis. Now, what is happening in this? Explain, I mean, you have to select which is wrong. Am I clear to you? Now, coming to that particular point, am I clear to you, children? It occurs outside the mitochondria. Am I clear to you? So, next one, what is happening, children? It is an anaerobic phase. Glucose undergoes partial oxidation to form 2 molecules of pyruvic acid, correct? Whereas glucose is phosphorylated to form glucose 6-phosphate. Am I clear to what is it getting converted into? Glucose 6-phosphate. Am I clear? So, neat and nodi answer na yadagirbodu. Actually, answer direct and the enzyme whatever they have used is isomerase enzyme. Adun talali itkoli. Am I clear to what is the enzyme they are asking you? They are telling you it is being provided by your isomerase enzyme. Is it so, children? Glucose getting phosphorylated, glucose 6-phosphate statement correct. but ಅಲ್ಲಿ ಇನ್ನೊಂದೇನೋ ಆಡ್ ಮಾಡಿದ್ದಾರೆ ಏನದು what is it they are telling it is an isomerase enzyme is it so are they isomers of each other no am I clear to you so what is happening over there makle here the enzyme was actually hexokinase what was it children it was none other than a particular hexokinase am I clear to you any doubt regarding this artha agtidiyandra so what is the option makle the option is nothing but your fourth one is D. Again, which of the following steps during glycolysis is associated with utilization of ATP? ಎಲ್ಲಿ ನಿಮಗೆ ATP ಯೂಸ್ ಆಗ್ತಾ ಇದೆ ಅನ್ನೋದನ್ನ ಕೇಳಿದ್ದಾರೆ. Am I clear to where and all you have your ATP utilized is the question. Now see properly children. Glucose to glucose 6-phosphate, correct? Fructose 6-phosphate to fructose 1,6-biphosphate, correct? Yes. So, 2 ATP molecules were actually used up in the preparatory phase. If at all you remember, in preparatory phase, 2 ATP molecules was being consumed. Yes, correct. PAP to pyruvic acid, no. Actually, ATP was generated. Yes or no? So, first optionally ATP got consumed. That is how you formed your ADP. Second one, fructose 6-phosphate, ATP again got consumed. That is how you formed your fructose 1,6-biphosphate. But PEPL ಏನಾಯ್ತು ATP was actually released. It was a payoff phase. ಇದರಲ್ಲಿ ನಮಗೆ ಡಿಫರೆನ್ಸ್ ಏನದು preparatory phase means this is a point where your ATP will be utilized. Payoff phase is a point where your energy will be released. So think PEP was in a preparatory phase or it was in a payoff phase. Obviously it was in a payoff phase. So definitely their ATP will never be particularly consumed. It will always be released over there. Where in the PEP. Understood. So what is the option? The option is D. Why both A and B is the correct statement. Am I clear to you? So the sixth one D both A and B. Now moving ahead. Sixth one. Which of the following? Am I clear to you? So coming back to the sixth one children, what is it? Which of the following conversions involve ATP synthesis during your glycolysis? Am I clear to you? Now, again, children, I am telling you, preparatory phase and your particularly payoff phase. Preparatory phase ali yavaglu ATP is being consumed. There ATP is being synthesized. It is always in your particular payoff phase. Am I clear to you? So here what is happening on that? The first option, glucose to glucose 6-phosphate, ATP was consumed. Fructose 6-phosphate to fructose 1,6-phosphate, the ATP was consumed. 1,3,5-phosphoglyceric acid to 3-phosphoglyceric acid. What happened here? Here, your particular ATP was synthesized. ಏನಾಯ್ತು ಮಕ್ಕಳೇ ಇಲ್ಲಿ ATP ಏನಾಗಿತ್ತು ಇಲ್ಲಿ ಮಕ್ಕಳೇ ಇಲ್ಲಿ ATP synthesize ಆಗಿತ್ತು ಯಾದ್ರಲ್ಲಿ the sixth question ಅಲ್ಲಿ. So the third one, whatever is there, am I clear to the third option? It will come under your preparatory phase or payoff phase. Obviously, it is in your payoff phase. So option A, the option is your C. Fate of pyruvic acid during your aerobic respiration. So pyruvic acid, if at all, if it is undergoing, your pyruvate is undergoing your aerobic respiration, what is the fate of it? Am I clear to you? Will it form lactic acid fermentation? No, because it is your anaerobic respiration. Alcoholic acid fermentation is also a particular anaerobic. Oxidative, definitely no. Am I clear to you? But oxidatively, yadu decarboxylation agtidya, phosphorylation agtidya aerobically. Obviously, children, it is not phosphorylation. Okay. Instead of that, there is a decarboxylation. Am I clear to you? So, seventh one allen agtide. During your aerobic respiration, there is a removal of carbon dioxide. Am I clear to you? So, what is the option for seventh one? Obviously, it is your C. Now, identify A and B in the given reaction. Okay, so identify what is A and B in the next question. Okay, so what is it? Pyruvic acid plus coenzyme A plus NAD acid. Okay, that's your nicotinamide. Next, in the presence of a pyruvate dehydrogenase plus magnesium 2 plus. What it will give rise to, children? What is A and B? Am I clear to what is A and B? Definitely A is nothing but acetyl CoA and B is nothing but your carbon dioxide. Yes or no? So what is the option for the eighth one? Again, it is nothing but your B. Ninth one. Select the wrong statement. Enu makla? Select the wrong statement. Now coming to the ninth one. What is it? Oxidative decarboxylation of pyruvic acid requires the presence of all the enzyme pyruvate dehydrogenase. Correct. All living cells, whether aerobic or anaerobic, perform glycolysis. Correct. Cyanide does not stop chemiosmosis. 100% it will stop. Okay, so the respiratory chain uses oxidation as the final hydrogen acceptor. So the option A no is your C. Substrate level phosphorylation, okay, occurs during which step of the Krebs cycle? Am I clear to you? So moving ahead, the next question is, what is it, children? The next one is nothing but your, it is telling you substrate level phosphorylation occurs during which step of the Krebs cycle anta kelidare. 10th one. So under this, succinyl CoA to succinic acid. And one more thing. What do you mean by substrate level phosphorylation? Where your FAD to particular GTP to GAP. Yes or no? So GDP. So ಅದೇ ರೀತಿ ಇಲ್ಲಿ phosphorylation ಆಗ್ತಿರೋದು ATP to ADP. Yes or no? So GDP to GTP, ADP to ATP. ಅರ್ಥ ಆಗ್ತಿದ್ಯಾ? ಸೊ ಇಲ್ಲಿ ನಿಮಗೆ ಫಾಸ್ಫಾರಿಲೇಷನ್ ಎಲ್ಲಾಯ್ತು ಅಂತ ಕೇಳ್ತಾರೆ ಕ್ರೆಬ್ಸ್ ಸೈಕಲ್ ಅಲ್ಲಿ. ಎಲ್ಲಾಗಿತ್ತು ಆಕ್ಚುಲಿ ಅದು? Succinyl CoA to succinic acid, isocitric acid to oxaloacetic acid, oxalosuccinic acid to alpha ketoglyceric acid, malic acid to oxaloacetic acid. So ಎಲ್ಲ ಆಯ್ತು ಮಕ್ಕಳೇ, ATP to GDP and GTP to ATP, GTP. ಓಕೆನಾ? ಸೊ ಎಲ್ಲ ಆಗಿದೆ ಎಕ್ಸಾಕ್ಟ್ ಆಗಿ ಮಕ್ಕಳೇ. ಸೊ ದಿಸ್ was none other than a particular succinyl CoA to succinic acid. So option ಏನು ಮಕ್ಕಳೇ? It is nothing but your A. The ultimate electron acceptor of respiration in aerobic. Yadro. Easy. Yes or no? So, the ultimate acceptor, the particular thing, who was it? It was none other than Elli water form agiddo. Okay? Water form agid yaar accept madiddu? Okay? Yaar accept madidru makle? It was your oxygen which accepted it. Okay? So, along with the proton, it got converted into your particular water. Phosphorylation of the glucose during glycolysis is catalyzed by, okay, so phosphorylation, 12th one, children, it was what to say, phosphorylation of the glucose during glycolysis is catalyzed by, where it is catalyzed by, children? Ellige glucose during the glycolysis phosphorylation, it was by your hexokinase. Yadu makla? Hexokinase. Glucose to glucose 1,6 phosphate ali phosphorylation alli agiddo. Okay, so that was none other than your hexokinase. Pyruvic acid, the key product of glycolysis, can have many metabolic fates under aerobic condition and it forms, what is it? Okay, children, so usually what will be formed, children? Pyruvic acid, the key product of glycolysis, can have many metabolic fates under aerobic condition. What it will form? That form agutte kandro, acetyl CoA plus carbon dioxide. Inu makla answer acetyl CoA plus carbon dioxide will be formed. 14th one. ETS is located in mitochondria. Helro avattu maths avagale maths of following madidvi now. Yes or no? So we did even the maths of following. So tell me, what is it? Membrane gottu. Outer membrane or inner membrane? 14th one. Tell me, children. The outer membrane or the inner membrane? Obviously, makla, it is nothing but your inner membrane. That is your C is the option. Which of the following exhibits the highest rate of respiration? So, yaadane nimge highest rate of respiration nadiyutte. Am I clear to you? In which of the things, children? Growing shoot apex, germinating seed, root tip, leaf bud. Now, here is the point. Shoot apex, germinating seed, root tip, leaf bud. Now, here is the point. What is it? In the 15th question, if at all you see, the growing shoot apex, definitely it is having a respiratory point. But the point is you also have a meristematic activity and they are already inactivate. But germinating seed, you will have what? You'll have a respiratory climax more. Yes or no? Lots of enzymes will be produced because the seed has to germinate now. Okay, so root tip and a leaf bud. Obviously, all the other things have a meristematic activity high. Am I clear to you? But in the germinating seed, there is also a requirement of respiration along with the meristematic activity. Am I clear to you, children? So the 15th one is nothing but your B. Select the correct statement. Okay, so here what are they asking? They are asking you to select the correct statement. Okay, pyruvate is formed in mitochondrial matrix. Is it so? Where is it formed, children? Pyruvate yellow ito Krebs cycle again. Yes or no? So cytoplasm, during which the conversion of succinyl-CoA to succinic acid molecule of ADP is produced. Definitely no. Yes or no? So oxygen is a vital in respiration for removal of hydrogen. Correct. So this is complete breakdown of glucose in the fermentation. Definitely not fermentation. Okay. So option is your C. 17th one. Mitochondria are called powerhouses of the cell. Which of the following observations support this statement? Okay. 17th one, what are they asking, children? Explain which supports the statement. Am I clear to you? Mitochondria are called as powerhouse of the cell. Which of the among the following statements support? Powerhouse anta ene karita idivi? Mitochondria synthesize ATP. Mitochondria have a double membrane. The enzymes of Krebs cycle and cytochromes are found in mitochondria. Mitochondria are found in almost all plant and animals. So out of all the four, which one is the one which is supporting your particular statement? Why is it called as a powerhouse of the cell? So then obviously, what is the answer? A. Why? It synthesizes the ATP, nothing but your energy. So that's the reason we usually call mitochondria as a powerhouse of the cell. The end product of oxidative phosphorylation. Okay, so the 18th one, which is the end product of oxidative phosphorylation. NADH, oxygen, ADP, ATP plus water. Yadu makle. So obviously yadrodu ATP plus water is the end product. Am I clear to you? Moving ahead. Match the column 1 and column 2. Okay. So select the given option properly. 19th one. Okay. So you have to match it properly. The 19th one question, children. Molecular oxygen is hydrogen acceptor. Electron acceptor is cytochrome C. Pyruvate dehydrogenase is acetyl-CoA. Decarboxylation is alpha ketoglutaric acid. So all these are the particular way of answers. So option yadu clean and model A. 20th question. The first 5 carbon dicarboxylic acid, okay, in Krebs cycle. Avagindanu helde nanu yadro 5 carbon compound yadidralli. Children, keep this in your mind. Talali itkomadi. Yadadu alpha ketoglutaric acid. Okay, so your alpha ketoglutaric acid is usually your 5 carbon compound. Arthakidendro, alpha ketoglutaric acid is usually your 5 carbon compound which is usually present in your Krebs cycle. Am I clear to you? And it is also used during your nitrogen metabolism. Maribedi. Okay, na? Moving ahead. Now, see children, identify X, Y and Z. Am I clear to you? What and all you have to identify? You have to identify the X. Am I clear to you? Always there is a problem with finding of X. And find out your Y and find out the Z. Am I clear to you? What is X, Y and Z? 21st question. Okay. What is X, Y and Z? So simple children, isocitric acid to alpha ketoglutaric acid. What does birth produce? NADH. Yes or no? So again to succinyl-CoA, what was the particular thing? Again it was NADH. Yes or no children? Now, succinyl-CoA to succinic acid, it was a GTP11 for oxidative phosphorylation ಬಂದಿದ್ದು. Okay, so fumaric acid ಅಲ್ಲಿ ಏನಾಯ್ತು, succinic acid to fumaric acid. If your FAD got converted into FADH. Am I clear to you? So what was the option? So option is your C. Second, match the following children. 1 with 2. Select the correct option from the codes given below. So 22nd. Am I clear to you? See the answer and tell me children. 22nd question. TCA cycle hands clap. Okay. And F0 and F1 particles ಏನೀವಲ್ಲ ಇನ್ನ ಮೈಟೊಕಾಂಡ್ರಿಯಾ ಮೆಂಬ್ರೇನ್ ಇರುತ್ತೆ. That is your ATP synthase point complex. End product is usually pyruvic acid. And pyruvate dehydrogenase is your oxidative decarboxylation. So option is ಏನು ಮಕ್ಕಳೇ A. Which step is called gateway step link or the link reaction in aerobic respiration? Okay, so which step is usually called as what ಮಕ್ಕಳೇ? Is usually called as a gateway step. So the 23rd question, which is it usually? The formation of acetyl coenzyme A. ಯಾಕೆ? This will link with your particular Krebs cycle. So it is your B. Next one. Krebs cycle is also called as your metabolic sink as it is a common pathway for. Okay. So Krebs cycle is also called as what makla? The metabolic sink. Am I clear to you? Why is it so? Children, carbohydrates, fats and proteins. Krebs cycle is also called as metabolic sink. Okay, because it is a common pathway for what and what? Carbohydrates, fats and proteins. Carbohydrates and fats only. Carbohydrates and organic acids only. Proteins and fats only. ಯಾವ್ಯಾವುದಕ್ಕೆ ಇದು ಕಾಮನ್ ಆಗಿದೆ ಮಕ್ಕಳೇ the Krebs cycle? Obviously, what is it children? All the three. ಯಾವ್ಯಾವುದು? Carbohydrate, fats and proteins. ಅದನ್ನೇ ನಾವು ನೋಡಿದ್ದು last ಇದರಲ್ಲಿ. What did you see children? In amphipolic pathway, everything was finally reaching where? Everything was coming back to your particular Krebs cycle only. Yes or no? So, hence it is called as your metabolic sink. Why? Whether it is carbohydrate, whether it is fats or whether it is proteins, everything is finally coming and sinking or linking to your Krebs cycle. So, option is your A. Yes, children. The last question. So, identify the enzyme A. Am I clear to you? So, what is the next one, children? Identify the enzyme A. Now, oxaloacetic acid plus acetyl-CoA plus water giving rise to citric acid and coenzyme. Which is the enzyme which is helping in the formation of citric acid? It is none other than your citrate synthetase. What is it, children? Citrate synthetase. Am I clear to you? Yes, children. So with this, we are done with the today's class. Thank you.